getting so many. Come back. Okie dokie. So chapter 10. This is the last chapter that the next exam is going to cover. Last time in chapter 9, we started talking about antimicrobials in general. And mostly chapter 9 dealt with when you have a contamination outside of the body, so in the environment, different things that we have, or when you're prepping a person for procedures such as, you know, surgery, injections, and things like that. And we looked at the different physical changes that you can bring onto the environment to cause either completely destroyed reproduction process, which is cidal. It causes, basically, we consider that as microbial death, or it was static, which means that it will slow reproduction down. However, if the conditions revert back to normal, the organism can start growing. So we looked at all the different ones. In chapter 10, we're looking at antimicrobial drugs. So this is things that we have to deal with microbial infections inside. First off, drugs. Definition of drugs. Drugs are any chemicals that affect physiology in any manner or the psychological changes. So all of those are referred to as a drug. Any chemical that brings on physiological or psychological changes in human beings. Chemotherapeutic drugs are drugs that act against a disease. So basically the everyday term we use for it is medicine, all right? But chemotherapeutic drugs are all these medicines that we can use in order to deal with a disease. I know we use the term chemotherapy a lot for basically cancer, but that's not just the only type of drug. It's just that they've tried to use that general term for it. But any medicine that is made up of a chemical, whether it's artificial or it's something that was found in nature, so synthetic or natural, is a chemotherapeutic agent or drug. So anything that even lowers like your blood pressure is under that. Antimicrobial drugs or agents, these are the ones that act against microbes and treat infections. Another term that comes with regards to microbial infections is a term that we sometimes use called prophylaxis or prophylactic measures. You need to know that term. This is a term when you use the medicine, the antimicrobial, against a specific infectious agent ahead of time of infection. So let's say you're going to Africa and you're going to an area where malaria is pretty prevalent. You use your prescribed anti-malaria drugs and you take it ahead of time so that if you get an infection, your body is ready for it with the concentration, high concentration of those drugs, so their reproduction will be low and therefore you won't get sick and your immune system goes against it. Vaccines and all of that are not prophylactic. Those are under different category. All right. A little bit of history, again, you don't need to know. Life without antibacterials, especially before the first antibacterial came about, that was penicillin, and then sulfa soon after that was produced in 1940s. So any little infection could kill you. This is the image of a little girl that got a cut on her face with Staphylococcus aureus, and then she could have died from it, but they had penicillin during that time in 1942, and they showed how she recovered. If you have a person that is around 90 years old or around that time, some of them were young around that age, or even things like saying a person has pneumonia would put fear of God in them. They were thought, okay, half of these people will die, because they would die. Like, they would tell you stories about their siblings and family, and oh, so-and-so, I had a brother, they died from meningitis. I had another sister, they died from pneumonia, and this and that. They would die very easily. So one little cut and you could die from it. And then after antibiotics were discovered, life totally changed. This is one of the reasons that a lot of people started taking antibiotics like crazy. It became the golden medicine. They would have a cold and they would take antibiotic. Anytime something happened, even though it was viral, they would take antibiotics, because they always thought, okay, if I get a cold, I might get bronchitis, I might get pneumonia. They wanted to prevent something, which in fact, not necessarily the case all the time, and they started overusing antibiotics. A little bit about how they realized that you can use chemicals, whether synthetic or natural, was the first person was this German guy, Paul Ehrlich, and he created arsenic against syphilis. And the concept came, arsenic, you know how difficult how dangerous that is. But syphilis was so rampant through Europe that they were willing to use anything. In some cases, it did work a little bit, some cases it did not. But he basically, from the gram staining, he realized different chemicals may affect different cells differently. So they were in the search of finding something that will affect the bacteria and not the human. That's why he called it the magic bullet. But Alexander Fleming was the first person that actually created, was responsible for creation of the antibiotic penicillin. He was a microbiologist. I don't know if you can see in the pictures, but his lab protocols and how clean he was in the lab wasn't one of the best. So he would have food and all that stuff here. He would have very old plates, new plates, everything combined. So he was not that organized and not that clean. One time when he was gone from the lab, he had a bowl of citrus next to some of his plates. When he came back and he was like, okay, let me look at, so the citrus had all this fungus, green fungus that we're used to on it, and that fungus is penicillium. Then he was like, okay, let me clean up and look at my old plates. What he noticed was on the old plates where it was totally covered by bacteria before, now he had the penicillium fungus growing on it. And around that, he was seeing clearing of the bacteria. So the bacteria were dying up and the plate was clearing off. So he was interested with that, and he started checking for what was responsible for this, and he found the protein or the enzyme, and he called it penicillin. He created a paper for it that this was what it's doing to a lot of bacteria. I'm testing it now, and it keeps doing the same thing on the plates, prevents them from growing. Then it came about these two guys, Howard Fleury and Ernest Chain, a British biochemist and a pharmacologist and a pathologist. So we are talking about after World War I, Europe was getting very big, big cities all over the world, a lot of infections going around, and people would die from simple infections. So what he noticed here was something very interesting. He noticed that they basically read the paper from Fleming, and they thought about it, and they're like, well, I wonder if it would do the same thing to a staphylococcus infection, for example, inside. So first thing they had to do is they had to try extracting penicillin, and then they had to make sure that they could purify it enough that it could be soluble in water so they can give injections to humans, things like that. Then the next thing was they had to purify it. They always knew this in some cases if you have never noticed, but a lot of fungal products, humans are allergic to them, or it can create an allergic reaction. Why? Because they are so close to us. So they wanted to test and see if the purified form that they had was good for use in humans. In the hospital, they actually had a woman who was dying from cancer, and she volunteered, and she said, inject it to me. I'm dying anyways, so if it's something very bad, you know, I will die, but it's okay, I volunteer. They actually did, she had a little bit of reaction. They went out and further purified it and created something that was very good for human use. Then they started testing on mice, and they noticed, hey, it works with mice. If you give it to them, and then give them the infection, basically they don't get the infection, but if they don't get the penicillin, they die from an infection. Then human testing started, and with that, this is the first guy that penicillin was ever tested on, Albert Alexander, police officer in England, and he had a cut on his face due to something, and he was basically dying from that infection, like he was losing an eye. So they started giving him penicillin, but realized this is the first time they're trying these things, and they had no idea about how much you have to give a person, how long. So at first he started basically getting really good, but then they ran out of penicillin. So remember, we have to take like penicillin for about a week or so. They ran out of it. I get it. So they ran out of penicillin, and he started getting worse and worse, and he actually died. It was really sad for them and for the family, because they noticed a change, and then he basically eventually died. So Flurry actually affected him very bad, and he said he promised that he would never do this again unless he had a room filled with it. It wasn't easy. They even tried to extract it from his urine, but they didn't get it. So next they started testing on children because it needed less dosage, and they did enough practices so they knew how much they have to give and things like that. But around the same time World War II started in Europe. So they came to United States and they asked for help. So they sent all the production to United States, and United States actually started creating these basically posters all over the world asking people to donate money for penicillin production. So they created the powder, and they would put powders, I don't think I have the picture, in wooden boxes with the old ways they had those glass syringes too. They used the same syringe on multiple people. With sterile water. So in the field, the medics would combine a little bit of the powder with a little bit of water and then inject the soldiers, and it basically allowed a lot of them to survive. Around the same time, this guy Gerard Dammack was a German scientist, and he was working on sulfonamide, sulfa drugs, or which we call now prontosills. And he realized that it has an effect on bacteria also. I'll tell you guys how sulfa, I haven't folic acid there, but I'll tell you guys a bit about these two specifically, how they work. But he was worried about testing on humans. So he was doing this during Nazi Germany, and failure in Germany at that time was not acceptable. So he was very worried. What ends up happening, his own child gets sick and was about to die, so he starts testing on his own child. And the child survives, publishes a paper, he creates the sulfa. This was the second antibiotic. And so he wins a Nobel Prize. Flurry and I forgot the other guy's name, Hardy, I think, and Fleming also, they get Nobel Prize. Fleming tried to reject, and he said, well, I didn't create the medicine, but they said, well, without your finding, we couldn't do it. He actually was not allowed to accept the Nobel Prize because of the Nazi Germany, but I'm sure afterwards, his name was mentioned, but anywho, I'm not going to ask you about the history, but it's nice to know these things. All right, so for antimicrobials, whether it's antibiotics, which are antibacterial, antifungals, antiprotozoans, antivirals, any of these, there is a key thing that in preparation of them or creation of them, all pharmaceuticals need to pay attention to. And that term is selective toxicity. Selective toxicity is basically this. Like medicine, when you say, you take an oath and you say, do no harm to your patient. Same thing is happening with medicine. You can't use a medicine that basically starts destroying the microbes, but at the same time it's so poisonous that it kills the healthy cells in the human beings. That basically defies the whole purpose. For certain things we have to go that way, like for cancer. The patient is dying because of their own cells. So that's why some cancerous drugs are very, they have a lot of side effects, and the patient almost sometimes says, you know what, I don't want the treatment, this is worse than the cancer. So, but we don't have anything, we are finding more and more things to get it specifically away. But so this means that the best medicine would be a medicine that selectively is toxic against the microbe, but leaves humans or mammalian cells, we are mammals, right? Our cells free and does not affect our cells. The more this difference is, the more toxic it is against the microbe and least toxic it is against us, that makes it a better and better drug. You can have this toxicity against structural differences, metabolic differences, and so on and so forth. Any new drug that comes out there, the research that you see, and also sometimes on the boxes you see what something labeled a number that says the therapeutic index. The therapeutic index means how selectively toxic is it. So the higher the therapeutic index, the better the drug is. That means it will have less side effects for the patient. So it will treat and target the microbe even better. So selective toxicity or selectively toxic, that's the term, and that's what they hope for. We're going to look at certain group of drugs, and I'm going to look at some that cause structural differences, some stuff they're microbiological or metabolic reactions, so we're going to look at each of those. Before I move on, why do I have a bacteria there? We've talked about this a little bit before in the last lecture or something before that. So when you think about antimicrobial drugs, when you are thinking about bacteria, funguses, protozoans, helminths, viruses, the ones that you go to a doctor and if you have an infection, they like, try this, if it doesn't work, I have this other one. If this doesn't work, I have another one. That's against what type of microbe? Do we have the most amount of variety of antimicrobial drugs against it? Is it bacteria? Yes. Why? Because they are prokaryotic. And they have certain differences with our cells, which are eukaryotic. Structural. They have peptidoglycan. We do not at all. Nothing that even comes close to it. They have a 70S ribosome. We have an 80S ribosome. Their metabolic activity is, in some cases, is totally different than us. For example, they have to make their own folic acid. We take it from our nutrients. We do not have any process, any biochemical process. So there is a lot of antibacterial. But when it comes to funguses, protozoans, and parasitic worms, they are also eukaryotic. So there is, the drugs that we have against them are very little. And sometimes their therapeutic index is not that great. So I don't know if any of you have ever taken an antifungal. Many times the doctor even warns you, you're going to have some side effects, it's going to taste weird, it's going to do that. And sometimes you just feel weird when you're taking it. Same thing with protozoans. Again, we don't have a large variety. With viruses, it's even worse. Why? They live in our cells. They reproduce in our cells. So how can you tell a chemical, this is a virally infected cell, attack that, this other cell that to you looks identical, leave it alone. Your immune system can do that, but a drug cannot. It just has a way of targeting it. So for those, we don't have a whole lot. Most of the antivirals are either preventing virus from getting into the cells, or if they are in the cells, preventing them from coming out, or sometimes they are slowing down the process of reproduction, but then it will have side effects on us. We would feel it too. So we'll go through all of those. Let's look at some of the structural differences. Inhibition of cell wall synthesis. When I go through these, I'll tell you guys what I want you guys to know. For most of these drugs, you don't need to know the name. Specifically, you need to know penicillin and sulfa drugs because they were the first ones. What's their name? What's the mode of function? For the rest, if you just know how we affect, for example, a cell wall. There's like 4 ways that we can have drugs that affect the cell wall production. So cell walls, inhibition of cell wall synthesis. This will destroy reproduction. Why? Because the organism that has a cell wall, when they reproduce and divide, the newly formed cells have to produce a cell wall and a proper cell wall. If not, then that means that cell is going to have improper cell wall. They are prone to osmotic pressures. They can actually explode, do this, do that. So they are not going to be a healthy cell, and they can very easily be damaged. Most of these medicines that have an effect on cell wall will have an effect on a newly formed cell wall. The original cell wall has been made, and in most cases, they don't have an effect on it. But as soon as the cell goes to reproduce and tries to make more and new cell walls, these drugs will inhibit that synthesis. So this is why for some of these drugs, you see that you have to take it for a long term, and you have to take it at specific times. Why? Because you want to make sure you target every newly made cells, and the concentration of the drug in your system is high enough that none of these can produce a good, proper cell wall. When it comes to cell wall inhibition, the microorganisms that have cell walls are bacteria and fungus. Protozoans don't have a cell wall. Helminths or parasitic worms don't have a cell wall. And virus doesn't have a cell wall, just uses your cell, right? So most of the drugs in this area are antibacterial. There is one that I'm going to tell you guys that it's antifungal, but other than that, it's pretty much focused on bacteria. Why? Because their cell wall has peptidoglycan, so we can target the reproduction of peptidoglycan. In any step of it, we can target that. So newly formed peptidoglycan. The first one we're going to look at is penicillin and its derivatives. So penicillin G was the natural one. Methicillin is a semi-synthetic. Cephalothin is another one. Cephalosporin. So all of these are basic derivatives of what penicillin is. So in your lab, we're working with penicillin G. So you have one of those discs that have it. Penicillin affects the cell wall and affects peptidoglycan. How the mechanism of affecting it is it has a beta-lactam ring. So this thing that they've labeled in green, that's what we call a beta-lactam ring. What it does, when you have peptidoglycan production, remember a peptidoglycan is made up of chains of alternating NAGs and NAMs, right? And those chains or strands of NAGs and NAMs have to be cross-linked to one another with a tetrapeptide. So 4 amino acids come and attach one NAM to the next NAM, and then you have few of them cross-linked intervals that creates the whole peptidoglycan. There is an enzyme called transpeptidase. That's the name of the enzyme. Bacteria have an enzyme called transpeptidase that is responsible for making these tetrapeptide crosslinks. The beta-lactam ring attaches to the active site of transpeptidase irreversibly. So it attaches to the active site of transpeptidase, not allowing it to get amino acids, put them together, and connect the NAGs and NAMs together. So a beta-lactam ring ends up being there with the enzyme. They can't create a chain or cross-link. So the chains are there. The NAG and NAM chains are made perfectly, but they're not connected. So basically, they're supposed to be linked to one another. They're not. Anytime the cell expands, these strands separate and the cell can explode eventually because of osmotic pressure or lose a lot of its water. So penicillin has beta-lactam ring, and that's how it affects it. And this is showing you how it can actually basically rupture. See, this is the old cell wall. This is a newly made bacteria, and it cannot make that cell wall that it needs to make in that area where it's split into two. So it has to cover the whole entire place. All right, cell wall synthesis inhibition. First was beta-lactam. So beta-lactam attaches to transpeptidase, prevents the cross-links from being formed. Vancomycin and cycloserine, again, don't need to know. Their effect is they specifically go and prevent, if they, in the peptide crosslink, there is an alanine, two alanine amino acids that connect to one another, they prevent them to make a covalent bond, or the peptide bond. Bactericin or bactericin actually prevents NAG and NAM production. Isoniazid actually is the one that we use against Mycobacterium. So those were the acid fast for TB and leprosy. It prevents them from producing the proper mycolic acid. So they don't produce a proper cell wall. 4 ways you can affect cell wall synthesis in bacteria. Either you have a beta-lactam ring and prevent transpeptidase from working. 2, you can specifically target alanine to alanine peptide bonds and prevent that from happening. 3, completely prevent production of NAG and NAM strands. And 4 is basically affecting in Mycobacterium, not allowing for proper production of mycolic acid. So if I ever ask you, these are 4 ways that antibacterials are affecting bacterial cell wall protection. Like I said, the others don't have it. Fungi have it. For candida infection, echinocandin is actually something that inhibits cell wall production in candida. But most of the times they are topical, so it doesn't get into your system and affect something that is similar. So that's one example. But mostly focus for cell wall on bacterials, antibacterials. The 4 ways that I told you, that's it. And penicillin, know that it's beta-lactam. The rest, you don't need to know the name of the antibiotics. Inhibition of protein synthesis. Again, in this process, if you look at it mostly, these are antibacterial. Protein synthesis in all eukaryotics follows the same rule. We use an ADS ribosome, we use RNA polymerase, our promoters are very similar. So the process is very, very similar in all eukaryotes. And again, a virus uses your protein synthesis process. So then it's very difficult to just target protein synthesis in one specific cell and allow it to happen in others. You'll see when we talk about antivirals, we have interferon that can actually do that, but I'll talk about viruses at the end. So protein synthesis, again, is mostly antibacterial. They will target the 70S ribosome. 70S ribosome functions the same as ADS, but structurally it's different. It is made up of different rRNA or ribosomal RNAs and different ribosomal proteins. So we can target those differences. I'm going to tell you guys about 4 most general antibiotics are in these 4 methods. Just want you guys to know these. Again, no need to know any of the names. Some people, it's easier for them to remember it with that, but some others, for them it's different. They just know the methods. So first way is by affecting the 30S subunit. So the small subunit of the 70S ribosome. Do you guys remember what was the job of the small subunit? Straighten it. So keep the mRNA nice and straight. These drugs, like streptomycin, gentamicin is actually one of the discs that we are looking at, is gentamicin. They have an effect on the small subunit's shape, so the 30S. In bacteria, it's 30S and 50S. They change the shape of 30S so it can't keep the mRNA nice and straight. The mRNA becomes bendy and the codons are misread by the ribosomes, by the large subunits. So it puts and misreads, they can't read it. So there's a bend. So for example, it reads that one, or it bypasses that one, reads these three as one, which they're not. So bending or changing the shape of 30S, one. Second way, they affect this one, the remaining three will affect the large subunit. There are enzymes like tetracycline, which again we are using in the lab. Tetracycline goes and attaches to the A site of the 50S subunit and blocks it. So the tRNA cannot come and read the codon. Again, that will prevent protein synthesis. In the other one, for the 30S, proteins they make, but it's going to be improper protein, improper codon. In this one, protein synthesis is going to stop because no tRNA can come and sit at the A site. Next one, again, 50S, but remember, the job of 50S was it had the P site, the A site to showcase the codons. After the A site and P site had their tRNA, what did the large subunit do? The next thing that it does, it's an enzymatic activity. When you have a tRNA with its amino acid at P and a tRNA at A, what is the next thing that happens? The peptide bond. Exactly. So that's the job of the 50S, the large subunit. Chloramphenicol, for example, affects that enzymatic activity of the 50S subunit, and they can't make the peptide bonds. So 50S is unable to make the peptide bonds. The tRNAs come, they think, oh, fantastic, everything is there. All right, my 50S is going to make a peptide bond. They cut the strands and the amino acids. The amino acids fly away. So you cannot create a polypeptide bond. You're reading properly, but you can't create the peptide bond. And the last one is it actually anchors the 50S to the ribosome. So erythromycin does that. It connects it there. It goes, the antibiotic goes and binds the 50S and binds it permanently to the mRNA. So the ribosome wants to move, it cannot move and cannot read anymore. Again, protein synthesis stops. So, 4 ways that I want you to know that protein synthesis and the process of translation is inhibited in bacteria. First one affects the 30S subunit. Prevents it from attaching properly to mRNA, causes mRNA to be misread. Second type, can go and block the A site on the 50S subunit. So tRNAs cannot read. Stops the protein synthesis. Third type, can go and actually prevent the 50S from forming peptide bonds. Again, proteins or polypeptides are not made. And fourth is by anchoring the 50S to mRNA, so ribosome cannot move and cannot read or showcase the other codons. Those are 4 ways that you affect protein synthesis or the process of translation, specifically in bacteria. Like I said, everything else is using ADS ribosomes. So we don't want to target it that much. Now tell me this. Are we completely safe from these type of antibiotics? Can they possibly have an effect, overuse of it in our cells? Why?
Mitochondria.
Yes, good job. Remember, our mitochondria have their own 7S ribosomes and their own circular DNA. So they have figured out over the time, they are doing a lot of mitochondrial studies, because there is a huge rise in diabetes and all these metabolic diseases that in the past the numbers were not that high. So one of the theories is this: they are thinking overuse of these antibiotics, like tetracycline for a while was the drug of choice. You sneezed, they gave you tetracycline. Didn't matter if you're pregnant, not pregnant, everything, you got tetracycline. Erythromycin, these were constantly being used because they're broad spectrum. They have an effect on a large group. And they are thinking that it is possible that it had an effect on the mitochondria. Mitochondria is something that usually the mother passes on to the child. So overuse of it, they're trying to see if that had some effect. There is a lot of research in this area. So they think it made our mitochondria not function as best as it used to. So there is a theory for it. All right, and like I said, viruses use our ribosomes. All right, next we're going to start thinking about cytoplasmic membrane. Can you think of much of a difference between cytoplasmic membranes in cellular beings, whether it's prokaryote or eukaryote? Did you notice a whole lot of difference? No, right? Really, not much. The only big difference is eukaryotes, they have a larger cytoplasm. They are still phospholipid bilayer, mosaic fluid mosaic structure. So we have phospholipids with proteins. If you can find a difference in the proteins in the membrane, maybe you can target that. But that would be a lot of research. You're targeting specific protein that there's nothing like it in anybody else. The other big thing was in eukaryotes, we have things like, for us humans, we have cholesterol in there. So maybe you can target the cholesterol or steroids in other eukaryotes. So not a whole lot, which that brings me to the example. For fungi, amphotericin B, that's a very common antifungal drug that we use. Amphotericin B targets ergosterol. This is something like it's very close to cholesterol, but in some fungi, instead of cholesterol, remember those fungi and plants don't use cholesterol. Cholesterol is an animal thing. So they have other sterols there. So ergosterol, they use that. Amphotericin goes and attaches to ergosterol and brings them together. So moves all of the ergosterol together. This is amphotericin. They attract each other and they create holes in the fungus. Most of the amphotericin Bs are topical. So we don't take it systemically. We don't want the high concentration of it in the blood, because ergosterol is similar to cholesterol. Too much of amphotericin in your blood and starts affecting your cholesterols and can cause damage in kidneys and livers. So they are just topical. It's toxic for consumption. Other than that, polymyxin is basically what you find in triple antibiotics. Neosporin is the other commercial name for it. Like if you go and they have a CVS brand, they write triple antibiotics, so they have 3 different antibiotics in there. They're good for gram-negative outer membrane, so they affect the outer membrane of gram-negative bacteria. But again, it's topical. Neosporin, you should not take it and ingest it. You should not even be using it for the inside of the eyes. Like if you have an infection, they say try to put it on the outside. Try not to get Neosporin on the inside. Too much of it damages kidneys, and too much of it damages our cells too. For Mycobacterium, M. tuberculosis is a very difficult bacteria to get rid of. So for some of these, we don't have any other choice. There is a very common drug we use for it, pyrazinamide, is actually one disrupts the transport across plasma membrane. If you've ever known anyone taking TB, drugs for TB, they'll tell you it's horrible. Like pyrazinamide can make you feel like liver enzymes shut up. The person who's taking it, they don't feel that great because it does have a side effect, but it's better than dying from, TB. So many times you hear in U.S., we'll talk about TB. In U.S., if you are like showing a positive skin test, but you do not have TB, like your lung, your sputums, your x-ray, everything is clear, they force you to take the medication for 6 months. And many patients stop about the second or third month because they say it was just too much. And sometimes even the doctor says, okay, enough, your liver is showing like the enzymes are not normal. But so cytoplasmic membrane would be something that I would talk a lot about, or I would have a lot of questions. But again, just know either antifungal, and you can target the ergosterol. Other than that, there isn't much. Plasma membrane remain the same for across all cells. And the viral envelope, they use our plasma membrane. They just insert their own viral proteins. Now we're going to go deep structural. We're going to look at metabolic pathways. In metabolic pathways, again, we don't have a whole lot. For some minor differences, we can find it, but it's not that much in the area. Heavy metals we can use, and some drugs actually have heavy metals. Most often these are for helminths, parasitic worms. One of the things about parasitic worms, especially we find the ones that we find in our GI tract, is they usually have a hook or a structure like a sucker, suction cups, things like that. They are basically, they attach to the lining of our GI tract, especially colon. Otherwise, when you have the bowel movements, they can be basically flushed from your GI tract. So they have to have ways of sticking there into the area. Many of the anti-parasitic drugs actually have heavy metals in them that paralyze the parasitic worm. So they won't be able to attach to the lining, and as you defecate, you just get rid of it. And they give it to you for a while, and then they like, you're clear. So that inactivates their enzymes and makes them feel paralyzed. One of the best ones that's antibacterial is sulfa drugs, although it's best because of its targets folic acid, but many people can be actually allergic to sulfa drugs. So this is sulfonamide or Prontosil that the Damac created. How this functions is the sulfa, the drug itself, is an analog of para-aminobenzoic acid. We call it PABA, para-aminobenzoic acid. This molecule is what in bacteria, then an enzyme goes, attaches to it, and changes it into folic acid. Sulfa is an analog of it, meaning sulfa drugs look identical to PABA. So the enzyme that is responsible for changing PABA to folic acid attaches to sulfa and cannot change it because it looks like it, but the inside structure of it is different. The chemistry of it is different. So the enzyme cannot do what it's supposed to do and cannot get rid of it. It's an irreversible interaction. So the bacteria cannot make folic acid. Folic acid is very important in nucleic acid production, and the bacteria that divides every 20 minutes needs a lot of folic acid. For us humans, it doesn't affect us because we don't make our folic acid. This is why a person, a woman who's pregnant or thinks about becoming pregnant, they are told by their doctor, start taking folic acid. For a fetus, they do a lot of cell division and all of that. They need a lot of folic acid. It's a group of B complex vitamins. So they have to take it a lot. Otherwise, the fetus will actually drain the mother. So for us, it's safe. So sulfa affects folic acid production. It's PABA analog and won't make folic acid, will allow the bacteria to make folic acid. Inhibition of nucleic acid synthesis. Remember, replication is one of the most important processes. Again, it's pretty similar to all cells. We don't have a whole lot. Here is just some minor differences. Remember DNA gyrase that coils and uncoils DNA for them, so it takes the supercoils out, and it can actually also, after the DNA is made, can allow it to go back and make the helical turns. We don't have that. We have topoisomerases. So quinolones or fluoroquinolones, you might have heard of these antibiotics too. They are prescribed many times, not as much as other ones, but they are there. So they inhibit DNA gyrase in bacteria. We don't have that. Rifampin binds to RNA polymerase of bacteria. We use that for tuberculosis. We don't use it for all of it because RNA polymerases still, they can be some similarities, but in mycobacterium can be used easily because we don't have the sequence of the same RNA polymerase as they do. Actinomycin was used actually to for antibacterial, and they saw that it actually has a lot of side effects because it binds to DNA at the open frame during transcription. So it attaches to DNA and RNA polymerase can't read. And then later on they realized it affects the healthy cells, so they actually now use it as a chemotherapy for cancer. Not a whole lot. Another one is antiviral here, AZT. Remember retrovirus, HIV is a retrovirus. Actually, discovery of AZT, I remember it in 80s. It was just like miracle drug. It really helped with progression of HIV. HIV is a retrovirus. So it has the enzyme reverse transcriptase. Think of your PCR. So the enzyme actually takes HIV, which is an RNA virus, takes its single-stranded RNA and makes a double-stranded DNA. That's how HIV goes latent in our cells. AZT goes and inhibits the function of reverse transcriptase, attaches to it and destroys it. There are purine analogs, acyclovir, herpes medication that a person who has genital herpes has to take it. If you are a person who gets oral, we call it oral herpes or cold sores. It is caused by a member of herpes family, is simplex virus type 1. It's different than genital herpes, which is type 2. But for some patients, they keep getting it. Doctors actually give them the same medicine. It's acyclovir. It has all these other different names, like the generic names for it.
Is that like canker sores too?
Yes, canker sores, cold sores, they're the same. So they give it to you, and the person who gets it notices that they're getting it because there's a tingling sensation. They're either a bit stressed, in a dry environment, something like that. And they will always tell you, uh-oh, I think I'm getting a cold sore. My husband gets them, so he's like, oh, I think I'm getting it. And the doctor has given him that same medication. As soon as he senses it, he starts taking that once a day. And the cold sore comes, but it's very tiny, not like the huge things, and it will go away within a few days. So sometimes they use that. What it does, it's an analog of purines. So the purines are adenine and guanine. So adenine and guanines are purines. It's an analog of those. So when it's trying to replicate the virus, it can't because it presses that. Again, for for nucleic acids, either it's like AZT, reverse transcriptase, doesn't allow them to do that. Acyclovir, which prevents the replication. We don't have a whole lot. This is actually where we have a lot of antivirals. Again, for viruses, it's so hard. How can you target the virus where it's inside your cell? We have some that prevent viruses from getting in. Tamiflu, amantadine. These are some of the antivirals that they use for flu or some viruses. Basically, it will prevent them from coming out. And this is why, for example, Tamiflu, they say it's too long. You're too far into the cold. It shouldn't have an effect. If you want, I'll give it to you, but it won't make an effect. But as soon as you feel like, okay, I'm coming down with something, and you know you've been exposed to it, if you take it within the first 24 hours, what it does is the flu virus goes inside of your ciliated cells, mass reproduces, it has a lytic cycle, comes out. These drugs prevent it from coming out. So they replicate, but they are inside of the cell. So what ends up happening in your body, basically there's low levels of them, and they can't come out and infect more cells. That gives your immune system enough time to target the cells that they are in and kill them and destroy them. And have macrophages, for example, remove those cells and then process with digestive enzymes. But if it's past that, already they've come out of the cells, your number of viruses have gone up, so not much of it. Fusion, this is something new that they have, and there's all these different names that are similar to it. This is the HIV one that blocks the entry of HIV into the cells. So these are the ones that you hear the commercial that I take care of myself. I make sure that I don't get infected. So if they have a partner that can be HIV positive, they can take this, and in many cases prevents the virus from going in so they don't get the infection. In general, for HIV we use cocktails, and the drugs have gotten so good that we don't say the person gets cured, but the number of viruses and infected cells becomes so low, we say this person is undetectable HIV positive, and at that stage they basically say they have such a low number of viruses that most probably they won't transfer the disease to somebody else. All right, characteristics of antimicrobial agents. Obviously, it's good if they are very easily made, available, inexpensive to us. That's important to pharmaceuticals. It's otherwise chemically stable. So if you can store it in any different environment, remember with the vaccines, the biggest problem was for COVID vaccine, it had to be like sub-zero, and higher temperatures destroyed it. So easily administrated, we'll talk about that in a bit. Non-toxic and non-allergenic to humans and selectively toxic. Spectrum of evaluations. So how much do they affect, what range of microbes do they affect? Efficacy. How much do you have to give it to a patient? How long do you have to give it to patients? Route of administration and safety and side effect. They always check these for a new drug. So spectrum of action. In the old days, remember, erythromycin, tetracycline, and sulfas, one of the reasons they still use that. They were broad spectrum. Erythromycin could affect gram-negative, gram-positive, and chlamydia, rickettsia. These are other groups of special bacteria. So they basically, whenever a person had an infection, they gave them that. They're like, you know, by the time we figure out what's going on, these will have an effect on it. And then there were things that were more narrow spectrum. In the old days, they thought broad spectrum was fantastic. But now we've learned when you use something broad spectrum, it affects your normal flora. They had no idea about normal flora, how important it is, normal flora. If you even have a little bit of another bacteria in you, but it's not growing, it's not causing an infection, it will kill that one. What ended up happening, it affected all bacteria, good and pathogenic ones, and it caused for increase in the number of resistant bacteria, which we refer to the resistant bugs and or superbugs. Super infections is basically when you affect a group of, your normal flora is just not one type of bacteria. It's a large group of bacteria and you have funguses. The broad spectrum affected a large group of them and then caused the other ones. Remember, these are opportunistic. We'll talk about them in more detail in chapter 11. But allows them to basically go about and like your other normal flora in your body to now start growing in number and cause a secondary infection in you. You've gotten rid of the first one, now it's your normal flora that you have to deal with, which is most cases is much more difficult to deal with. Efficacy or efficiency of it is basically measured by disc diffusion susceptibility test, minimum inhibitory concentration, MIC, and MBC. So disc diffusion, we talked about it in the lab. They can actually see whether or not it's susceptible, resistant to it. What is the percentage? They can create serial dilutions of a drug and see, okay, if you have this many grams of it, what it does. But we'll talk about that. Most pharmaceuticals, this is the first test that they do. We call it MIC, minimum inhibitory concentration. So same thing that, remember we talked about dilution test that we did for the reagents, they do the same thing. So this is an image of tetracycline. So they create solutions of tetracycline, different concentrations, 0, 0.2 micrograms, 0.4 micrograms, 0.8 micrograms of tetracycline in the media. Then they add the bacteria of the interest to it. So 5 drops from the same culture to all of the tubes. They incubate and come back and look at it the next day. Any tube that has no growth to it, that first tube, they say that has the minimum inhibitory concentration. So in this case, for example, 1.6 microgram of tetracycline inhibited the growth. The others, see, they're cloudy, so they grew. They were not affected. Then they do something else. They take all of these clears and take a sample from them and put a drop on a fresh plate without tetracycline. What they're checking for is minimum bactericidal concentration, MBC. Out of those tubes, was the inhibition just like in 1.6? Was it inhibited? And as soon as you take the drug away, they recover. So was it static or cidal? And from that, they can tell you, all right, among these, this concentration is bactericidal. So it will never recover from it. And based on this also, they provide all these like, all right, you need to take it 3 times a day for 7 days or for 5 days and things like that from those tests. Routes of administration. So this is also like, obviously anything that you can say, take 2 of these and call me in the morning is the best. Why? Because the patient knows, all right, put these, drink some water, swallow it, and that's how you're going to take it. So there is no special way of doing it, or even topical. However, these graphs are showing you the concentration in your system. Systemic meaning in your blood. Topical basically should not get into your blood. I know there are some creams that they give and they're like, okay, use it in this area. Certain areas of your body, there's more blood in it, so it can absorb it. But you put very highly concentrated. Antimicrobials are not like that. So in most cases, when you use topical for a short amount of time, it doesn't get into your blood. For the oral ones, pills, the other liquid forms, it goes into your system. Some of them sometimes are absorbed right directly from your epithelial cells that get into your system, and the others have to be digested. So you usually see a tiny little spike on your in your blood, but this is why when you take oral medication like antibiotics, you are told to take it exactly at the right time every day for this amount of time. Why? Because you take it an hour later, that concentration has dropped. So you might give the bacteria some chance to try to recover. This is why they say stick to it for exactly at the same time. Same thing with birth control pills. You don't want that concentration to change. If you're taking it in the morning at the specific time, always take it there. Not like, okay, today I'm going to take it at noon, tomorrow is, you can cause it not to work. But if you keep it the right way, it should work. Some drugs are good for oral, some others you can't. You actually, the concentration would never spike. So those are when you get intramuscular injections or intravenous injections. Intramuscular is injected into your interstitial fluids, which is absorbed by lymph, and then from lymph gets to your blood. So it becomes systemic, and again, with a simple injection, immediately it can spike up there. And if you keep getting injections, it will go there. Intravenous, obviously, for a infection that is very hard to get rid of, this is what they do. If you've seen, you might have seen it, some people who have had a staph infection, which they're worried about it, they send them home for 6 weeks attached to an IV. They're constantly getting antibiotics. So it spikes up, and as long as you're attached to that IV, your concentration is really high. But again, the last two, you need to teach a person how to give themselves an injection. This is, not everybody can actually create an IV and inject and all of that. So, and finally, toxicity. So selectively toxic is very important. Whenever a new drug is out there, if you can, don't take it for a year or two. Sometimes you have no choice. But usually, sometimes, even if they basically were, the research that they sent out for FDA to accept it, if it wasn't completely 100% honest, within 2 years or so, it will start, if it has very toxic and it's not good, you will hear of people that have kidney damage and liver damage. So that's usually where it goes. Also, the difference is pregnancy. There are some drugs that they say it's perfectly healthy for you, but because you're pregnant, you can't. It will affect your fetus. A fetus is much smaller, it's constant development, so the concentration that for even a child might be okay, for a fetus is not. So you have to consider that. After that, most often it's allergic reactions. So minor, typically that's okay, some side effects. We don't want to get any anaphylactic shocks. And disruption of normal flora also. I am standing up. My watch is telling me stand up and start moving. I'm not dead. All right, it just wants me to do this. Development of resistance. So remember I said we'll talk about this in more detail because we're obviously more worried about this when it comes to our medicine. First of all, resistance, where it came about, the cellular appearance of it. So the biochemical reactions or the cellular existence of resistance, where it came from. Typically, the cells or the microbes were naturally resistant to it. So in a population, even in bacteria, you could have a wide range of genetic frequencies. So it is possible for some of these bacteria that naturally at the beginning, something had happened, and let's say if it affects their protein synthesis, for whatever reason, their 50S in this group of like 1 or 2 of the bacteria in E. coli, the strain that you were using for, their 50S was slightly made differently because of some genetic frequencies or something that happened. And they were naturally resistant to tetracycline, let's say, okay? But majority, about 98% of E. coli is susceptible. With overuse, what ends up happening is the number of resistant can increase. So naturally, they had that resistant in those few. Or sometimes partially resistant, completely or partially resistant. Some of these, the tetracycline worked on, but you needed, instead of 7 days, you needed 10 days of tetracycline. So once you stop it, that one recovers. And they're like, ooh, okay, I can handle more tetracycline next time. So naturally, it was there. It wasn't like some mutation happens after ages. No, in most cases, the naturally is there. Again, how it came about naturally, it was, there were mutations. One bacteria has a different gene. How they pass the genes together, all of those can happen. How it's passed on is, again, either by natural ways, why it increases in number is by natural ways, and then I'll come back to this. So showing you this image. Remember I said the 2 red ones here are the ones that were naturally resistant to begin with. The first time you use tetracycline, they were actually doing this. And these 2 were resistant. The more times you use it for the same microbe, what ends up happening, the ones that are susceptible will die. These 2 now have less and less resistance. They keep dividing. They keep surviving and dividing. Then what ends up happening is you have a population that now is mostly resistant. This is called bottleneck effect. So you get rid of those, now these ones are the ones that we are growing without competition. Before, the others kept their numbers low.
Does that resolve like C. diff in our normal flora?
Yes, because so C. diff or Clostridium difficile is the one that we now worry about in hospitals. They kept using so many antibiotics, and some of them were naturally resistant. So they kept dying, and the ones that remained are now resistant to a lot of antibiotics. That's why for some C. diff infections, they can't actually use antibiotics. It won't work, so they do reverse, they fecal transplants. So they can increase in number that way. These guys also can spread it to other bacteria by processes such as transformation. If one of them dies and that resistant gene is there, another bacteria can pick it up and now they have a plasmid. Transduction, conjugation, they can pass it on these ways. How do they resist? Like different ways that they can resist. Some of them actually have a pump that pumps the drug out. Some of them have an enzyme that goes and destroys the drug. So these are different ways of resisting it. Altering the target of the drug. The target of the drug is altered. It's different in them. So the drug can't actually figure it out. Metabolic pathways are different in the group that are resistant. So these are some possible ways that they show their resistance and helps them survive the drug. How do we, other thing about resistance. You say something is resistance, is cross-resistant or multiple resistant. Cross-resistant is something that, okay, if it's resistant to penicillin, it might be resistant to penicillin derivatives, like metacycline and not methicillin, methicillin, which is derivative of penicillin, a synthetic form of it. So they might be across the same family of drugs. Multi-resistant ones are worse. Those are the ones that are resistant to multiple different kinds. So penicillin, tetracycline, erythromycin, multiple ones. TB, I always worry about TB because in Africa now we have TB that is multi-resistant to many drugs. They have nothing to take care of that TB. And right now it's in those locations. If it spreads throughout the world, it's disastrous. Common places that you see resistance appearing, hospitals and nursing homes. Because there's a large group of infections, different kinds, large group of different drugs, and with the processes of, again, transformation, transduction, things like that, they can actually exchange material. These result in what we call nosocomial infections. We'll talk about it in more detail on Wednesday. Nosocomial infection is an infection that a person goes into the hospital with something and comes out with a different infection. And also has caused resistant or superbugs. How we are now trying to prevent resistance. Limit use. If you have a cold, don't take an antibiotic. Unless by a doctor you've been told that you are prone to, you have lung damage, so this is a very bad cold or flu, let's start giving you an antibiotic. Your oxygen levels are low. If you're prescribed, finish it and take it perfectly how you're supposed to take it. Use antibiotics in combination. So they are trying to, for some of them, like for some ear infections for children, where they have a resistant bug, they give them 2 antibiotics together. But again, here, with antibiotics and your other medications, always get them from the same pharmacy. You have to worry about synergistic and antagonistic relationship. So for example, antibiotics disrupt the function of birth controls. Many forms of antibiotics. So your pharmacist might be able to say, by the way, protect yourself for this month because this happens. Or you can have a patient that comes and says, I have no idea what happened, and then you ask their history and like, yeah, you were prescribed this antibiotic, so it could have an effect on your blood pressure medication, things like that. And development of new variations, 2nd generation, 3rd generation. Usually they don't give this to general public, they only give it to the resistant, the public that the individuals that have those. All right, and that's the end of it. Okay. That one says something. What? According to 2? No. All right, it's almost 1 o'clock. If you want, we can take a break, and then I can go over a little bit of review, or like just like a few minutes. If you want to go stand up, wake up, and all that. Then I'll just go through again the chapters and remind you guys what to study, so you can get started with the studying, or if you have questions. All right? Okay. And I'll stop recording.